Can't even check in here. All right. Welcome, everyone. I am here finally at Ginkgo Bioworks, leading with AI and automation. You guys have already seen the paper that I posted earlier today on linking ChatGPT with their cell-free assay. And so I'm here live in person, and we're going to get a tour. Here's my tour guide today. Hello. Please introduce yourself.
My name is Rachel Machete. I'm one of the co-founders here at Ginkgo.
Thank you. All right. I'm off to go. Let's go.
Okay, awesome. So this is our rack installation here in Boston, Massachusetts. It's about 43 different racks, 55 instruments on there. And actually just even at the end of December, it was only about 36 racks. So we've been rapidly expanding it to add new capabilities, add more capacity. And our goal is to be at 100 racks by the end of May. And so really, the distinguishing thing about this particular automation setup is that we have really built it from the ground up to be focused on what we call autonomy rather than just automation. So autonomy means that this system can concurrently run many different workflows all at the same time, right? So most integrated automation systems, people have a particular workflow in mind that they want to run, and then they design the entire integrated automation system around that particular workflow, which means that if your workflow changes or you want a new endpoint readout or whatnot, you kind of design and build a brand new system. This is intentionally designed to be able to run many different workflows. And so when we originally built this technology, first at Zymergen, but since further developed here at Ginkgo, we essentially thought about racks as really being a device-centric design. What that means is instead of designing the whole system around one workflow, we really think about what are the set of instruments, what are the set of devices you want to use in the lab to conduct your experiments of interest. And then it's a question of, well, what are all the different workflows that set of instruments can support? So every rack here essentially contains one instrument type.
And what you call a rack is one unit?
Yeah, a rack is essentially a reconfigurable automation cart. So each of these physical boxes is a different rack. You can see some of our older generation racks here on the left, and over there on the right side you can see some of our newer generation designs. So we simplified the number of parts, kind of made it look a little nicer in our latest generation design. And each rack contains an instrument or maybe a couple copies of an instrument. So for example, that one has thermocyclers in it, has a couple thermocyclers. You can even go up to 4 thermocyclers in an individual rack. And so you should think of each rack as essentially containing an instrument, and the rack itself is this hardware wrapper around that instrument designed to make it operate more reliably and hook it into the overall automation system. Connecting each of the racks is our sample transport track. That's essentially railroad tracks that can have little train cars on them, and those train cars can transport SPS formatted plates between any instrument. So when you write your protocol, you just say, well, I want to go from this instrument to this instrument to this instrument. You can also specify the parameters with which you want to run each of the instruments in your protocol of interest, and then you can essentially run your experimental workflow. So in addition to the hardware wrapper that is the rack, we also essentially write a software wrapper around each instrument. So we've developed device drivers for every instrument on the system so that all the control you would have if you were just a bench scientist using that instrument in the lab, like going up to the PCR machine and programming your PCR protocol, you could also do that through our software interface. So we have built a software called Catalyst that essentially allows you programmatic control of all the instruments on the system.
Now, what happens with errors? Like, things get buggy all the time.
Yeah.
I know PCR machines, so polymerase chain reaction machines. This is typically used for cloning. If you know PCR tests from COVID, then you know a thermocycler is used for PCR machines. And so let's say, there's an outage, and that is like, maybe number 2 step within the full automated system.
Yeah.
What happens in that case?
Yeah, absolutely. So if an error happens on the system, we actually have a camera inside of every rack, and that camera, and automatically when an error happens, the last kind of 10 seconds or so of camera footage is saved. And so automation engineer, even a remote automation engineer, can actually log in, look at the error message, and even review the camera footage and decide, well, how do I rescue this error? Sometimes it's as simple as retrying the same step over again. It just happened to not work, but if you retry it again, it'll work. Sometimes it requires, aborting the step and restarting from scratch. So it kind of varies depending on the error, but the point is you get our support team gets a notification of the error. They can go in, figure out what's going on. If they need help from somebody who's physically next to the system, they can ask somebody to come in and help rescue the plate and whatnot. So, but we find about 80% of errors can actually be rescued remotely with our system. And so for folks who want to buy racks and use them on site, we can actually provide that support ourselves for you as part of the support we provide. Or if you want your own automation team to be providing that online support, we can train your team on how to do that.
So is it running 24 hours and manned 24 hours? Or?
We don't quite have 20. We do run 24 hours because people could be running at any time. Our support is more like 20 hours.
Oh, nice. 20 hours. That's amazing.
So
oh, wow.
Our first shift starts at 6 a.m. in the morning to start supporting the racks, and it goes until I think 3 a.m. or so. So I guess 21 hours technically.
Wow.
Yeah.
Okay. So the robotic arms are taking everything from instrument to instrument based on the software that's outlined by the code.
The arms transport plates from the instrument onto the sample transport track, and then the sample transport track can move plates between the different instruments.
And those are those tracks right there.
Yeah, the silver things that kind of look like railroad tracks. That's the sample transport track that can actually, you can see some samples moving over there. They just moved in front.
That's a plate right there.
Yeah, that plate's running by, and you can see it's kind of transporting there. So it's moving and it's going into the particular instrument that's up next for that particular plate.
And so the plate is what's customizable right here. So you can go from like 96 to 384, even
so we support
culture plates.
Yeah, we support a variety of plates as long as they're in SBS format. So 96 well, 384 well, you can even do 24 well, 6 well plates.
Just depends on the experiment.
Yeah, it depends on what you need for your experiment.
All right. Maybe we can just go through a gamut of like just some of the experiments that you have run already.
Yeah,
just like
absolutely. Yeah,
we already say PCR.
Yeah, so we run everything from like NGS sample prep, next-gen sequencing sample preparation, cell-free protein synthesis, obviously, given our paper recently with OpenAI. We can do culturing of cells. We can do protein purification. We can do DNA assembly from oligo pools. We can do protein quantification assays like high-bit or simple absorbance measurements.
Any biophysics? Like DSC or CD or?
Yeah, so we have qPCR machines, so there's certain things you can do with like, looking at like melting temperatures.
Yeah.
And stuff like, do those.
Cool.
So it's really not constrained based on the science, it's really constrained by the instruments that we have on the system. So anything that you can run using, typical liquid handlers, PCR machines, plate readers, bulk liquid dispensers, incubators, shakers, qPCR machines. I might be repeating myself at this point, but any workflow that needs one of those, you can essentially run on the system.
And for the protein purification portion, from the paper, I think you're doing it through magnetic beads, right?
Yes, we didn't do protein purification in the paper, but we can do protein purification. It is magnetic bead purification.
Okay.
Handler.
And so are you able to do like secondary purification? Is there like an ACTA system hooked up with any kind of like?
We don't currently have an ACTA system hooked up, yeah.
That's in the future, maybe?
Yeah, that's on the roadmap, yeah.
Because I could easily see this connected to like a crystal farm, and you could even, connect to a mosquito and or have the leaf.
I mean, there are papers that have been come out about using echo liquid handlers. You know about echoes?
No.
So the Echo is an instrument made by Beckman. It's what's called an acoustic liquid handler.
Okay.
Essentially, it's capable of transferring very, very small volumes of liquid. So the ones that we have on this particular system can transfer as low as 25 nanoliters, but there's another newer version of the instrument that can go as low as 2.5 nanoliters.
Nice.
And so you can do very small liquid transfers. It tends to be very useful for compound screening, but people have also used it for crystal screening of conditions for forming crystals. X-ray crystallography.
Okay. So Echo, and then I guess some of these are Hamiltons and Tecan.
The particular liquid handlers, we have Agilent Bravos on there. We have a Formulatrix Flow i8. We have Echo acoustic liquid handlers. We also have some bulk dispensers like the Templist and the Multiflow.
I see. And so if an experiment requires like an overnight incubation, or maybe even, there are some experiments that require like 3 days, 72 hours, something like that.
Yeah, that's fine.
That's manned by the scientists.
No. So incubation can be done on the system too. So we have a Cytomat there, which is a incubator, if you need like shaking with humidity control. We also have a rack with 6 Ginkgos on it, which can do individual plate shaking, no humidity control, but temperature and shaking control. And then we also have the Stereosaur, which can do static incubations.
Wow. We're not going in, right?
We are going in.
Oh, we are?
Oh.
Okay. I was worried about sanitary. We're going in, you guys. Go check it out. And so I know so there's one idea that potentially, like, any scientist anywhere in the world can remote access and basically design the experiment and be able to make it run.
That's the eventual dream. We're not there yet, so we don't provide access to the lab to anyone. But we are. So we've been sort of expanding our offerings from. Originally, we started with kind of end-to-end R&D projects that we did for customers, and then more recently we launched our data points offerings, which is sort of a menu of CRO services where we can do data generation for customers. So things like functional genomics, antibody developability, and ADME assays. And then we're now looking at expanding to more and more like what we call kind of cloud lab services, where, for example, if people just want to be able to measure expression or measure or get purified protein back. So they give a sequence, they get purified protein back. We're planning to launch that in a few weeks. So.
Wow. So right now it's mostly startups or biotech startups, or what's your client base right now?
Yeah, mixture. There's big pharma, big ag, and then as well as biotech.
So basically people who want to outsource this part of it. They do in silico portion and then.
So different people want to do different things, right? Some people want to outsource the entire R&D project. Other folks want to just get some data for a particular project that they have that's going in-house, and they just want to pay us for data gen that they then own the resulting data. Other folks want to be able to do the research on-prem, and they actually just buy the racks themselves so that they can actually run their own experiments in-house.
That's exciting.
Yeah, so we try to sort of meet people where they are, whatever you want. You want to outsource it, or if you want to insource it, you can do so with this technology.
And for the people who are buying racks, you're actually installing on site with them and helping them set up and run.
So, for example, Pacific Northwest National Lab, we just opened a new facility built on top of benchtop racks. They actually wanted to be able to do phenotyping of microbes, and in particular anaerobic microbes. So we built the entire system inside of anaerobic chambers for them. So it's about an 18 rack system, I believe. And they actually, on the basis of that, just recently awarded a contract to Ginkgo to build an expanded 97 rack system. So we're going to be building that for them soon.
That's amazing. Okay, cool.
All right, so you want to put on your lab coat?
Yes.
You grab a lab coat there.
All righty. All right, I'm going to put on a lab coat, you guys. I have to put you guys down for a second. Let me put you here. Is that okay?
Yeah, you can put that there.
You guys can continue on Instagram or Twitter or YouTube or Facebook. Know that on Instagram I have the glasses on, so you're getting another view there. Hopefully I'll splice it up and you guys can see both views in the final. And leave this also on the rack.
Yeah, you can hang it on the coat rack or on the shelf here.
You can see us. I think the futuristic world of having any scientist log in and run an experiment, that sounds so cool. It sounds like the grounds of like citizen research, citizen science research. Yeah. Okay. All right, we're back. We're all suited up, you guys. Let's see, maybe you can see me. I'll do front camera quickly so you can see me. We're in the lab. We're in the future. All right.
So here you can see a little plate on the track there, and it's kind of zooming around. There's an arm that's
gosh, you guys, we're literally in the future.
equal plate moving around the system.
So that's the tracks, one plate going from one instrument, going to another one, whether it's assay or to a new experiment. And then that's the robotic arm. And they're all encased in here, so sterile. Or at least like BSL-2, I guess, is it? BSL-2, yeah. experiments can you estimate are running right now? Oh, oh wow. Oh,
So
they
this is
got a
our
track.
Catalyst software. So this is showing what's actively running on the system right now. Each of those rows is a different instrument on the system. The colored blocks, each color represents a different protocol run that's running on the system right now. So filled in colors means that particular instrument is occupied by that particular protocol. So you can see here it's getting cut off, but you can see all the different protocols are running. We're running some self-reproductive synthesis, we're running some liquid handling, we're running some plate reads, we're running some DNA synthesis protocols all at the same time here on the system. So those are all being run concurrently. We have about 12 different protocol runs all running on the system right now. In a typical day these days, we are running 80 to 90 protocol runs in a day, and it usually consists of about 30 unique protocols that are being run.
Because you're doing things in triplicate, replicate.
Yeah, some things you're running more than one place, so that would be multiple protocol runs, but averaging kind of 25 to 30 unique protocols being run every day. Our goal is to get that number up to 80 by mid-year.
That's cool. Without talking about price points, how are people paying for this? Are they paying per protocol run, per experiment design, per like how? What's the distribution?
So it depends on how people are accessing our services. So for an end-to-end R&D project, it would be a fixed fee. We're running some of those on here. For some of our data generation services, it would be basically per sample cost.
Per sample cost. Okay.
Yeah.
And mass spec on site?
We do have mass spec. The mass specs are just over there. They're not yet integrated on the system because mass specs are pretty robust. They can run, for 36 hours at a time if you need to. So we do the sample prep on the system, but then actually haven't yet integrated the mass spec itself.
Nice. And so is each of these instruments integrated into that? Essentially, is your lab notebook essentially?
Yeah.
Is that an ELN?
That software that you just saw, that's essentially tracking everything that's running on the system. So it's reporting when stuff starts, when stuff ends, any errors that happen. We track like the temperature at which instruments are being maintained. If a liquid handler has a transfer error, it'll log that. You can essentially keep track of not only the experimental data you're generating, but the metadata on how that experiment was run is all being recorded into our databases.
That is really cool. And then what's the human intervention between observation from the instrument versus final lab report?
Yeah, so basically that can either be done by humans or it can be done, via AI. So, the data gets logged into our Snowflake instance, and then humans can go in and analyze that data and plan the next set of experiments. Or in the case of what we showed with GPT-5 driving the autonomous lab, we were essentially feeding the data back to the model, and the model was deciding on the next experiments to run. So we would run things kind of in batches of like 16 or 32 plates. 16 or 32, 384 well plates. So that's a lot of data.
That is a lot of data.
We would pass that data all back to the model. The model would crunch it and then decide what are the next set of experiments to run. So it can be either an AI in the loop or a human in the loop with an autonomous lab. Like an autonomous lab can be driven by either one.
And so now where does agentic scientists come into play here? Like, is it a situation where I can imagine each one of the instruments will have its own agent maybe?
It's a good question where things are going to go, but what I sense is so part of what we've enabled with our Catalyst software is it allows you to programmatically specify multi-instrument flows, right? Multi-instrument protocols. And so when you're developing that protocol, right now it can be in the case of our pre-print with OpenAI, it was humans who wrote that protocol. Yeah. But you can imagine just as well that an agent could write that protocol, an agent could debug that protocol, and make sure it's right. And then you'd have another agent who might design the experiments that use that protocol, right? So just like you have different members of the team, different human scientists who might do different parts of the work, you can imagine different agents that are good at data analysis versus design of new experiments versus verifying that protocols are work the way you'd expect before you run them. And so it could be a team of agents who are working in conjunction with a team of and a particular experimental objective met.
That is so cool. I will not stop saying that. It's so cool. Because it seems like you kind of started with a grad student and PI as an example of your OpenAI platform, where the humans are kind of the PI supervising, and you have maybe postdoc actually, I would say, as your OpenAI designing and analyzing the data. And it seems like now you're moving towards exploding the team to a multiple research scientists and senior scientists.
I think what folks giving models access to not just what's in the context window, but actually tools. So giving them access to computer terminals, to data analysis packages, giving them access
Claude.
to the internet, etc. That makes the large language models much more powerful. And then if you could also have a team of models who are working on different aspects of the problem, like a team of agents that are working on different parts of the problem, working together to figure out the best answer collaboratively. You have, just like humans working in teams are oftentimes better than an individual working alone. A team of agents might actually be a more effective way to plan and execute science than a single agent alone.
And so the protocols you're running here, are these being written by a Claude bot or pre-installed? Like,
right now most of the protocols have been written by humans, but I can that protocols could be written by agents as well.
Okay, I didn't even think of this. Trash.
Oh my
Is human done or?
Let me show you.
I didn't even think of that. That is cool. Hello, sorry for disturbing you. Oh, wow.
So my favorite run on the system is actually on the other side here. 1 second.
Okay, sorry.
My favorite rack on the whole system is what we call a trash rack.
Oh.
Basically for throwing away the trash from an experiment, right? So use tips, use plates, they can get dropped into these sharp spins, right? Because you generate waste when you're doing experiments. And so you can see we have a little camera above each of the 3 bins, so it can detect when they're stop filling and call for them to be popped out. It'll actually also randomize the location in which it drops the plates in the tip box, so that you don't just end up with one stack of tips or plates all in one location. You want to be able
to maximize the space in the trash.
Filling the trash with plates.
That is clever.
So why waste space in a storage rack for your used tips or anything? Why not have a trash rack for disposing of your waste?
That's really cool. And so the cameras I think detect if the trash is full, but do they also detect if, like, say the plate still has stuff and you shouldn't be thrown out? Like, like they could be mistakes.
That level
No,
of
okay.
resolution. Yeah. So assuming that they thought about whether they could be trash
And reservoirs also treated.
there's not run reports in the system actually because of spillage like that. So we typically for well played or
You're stamping.
or you go.
Oh, like the
So
aspirating multiple
this
blocks.
is multi-flow, and so you can do like bulk dispense of reagents, like this is a phosphate buffered saline, for example, and you can do a bulk dispense if you need to dispense the reagents in bulk.
Now, so what's an example of like an open loop, like experiment? Like I'm trying, because that's what I've been trying to wreck my brain thinking, like, because you're iterating so fast. Ideas have to come also so fast. Like, I can imagine a situation where you are like changing the protocol in the middle of an experiment, but you probably can't do that, right?
Midstream, you could. It's a little tricky to do that. So typically we would change the protocol between different protocol runs rather than in the middle of the protocol run.
And when you said it catches errors and then restarts and reboots, it's catching the error, or the scientist who's manning has caught the error and is able to reboot remotely?
Yeah, so right now typically on the system that is monitored by our remote support team, and then they will try to basically continue the protocol on if they can rescue the protocol run from the error. However, one of the things that we're looking at is, for example, when you use these echo acoustic liquid handlers, they can generate what are called exceptions, basically liquid transfer failures. There's no reason you can't take that exception log, right, and then actually use that to continue to fix this. So you could just kind of rework it as part of your protocol. So we're
that is cool. That is really cool. Wow. And then, all right, so all of this is in vitro. Obviously, it can only be done in vitro.
We don't use like animals on the
Right.
system, yeah.
Well, yeah, obviously. But I'm just trying to think of like what are some futuristic examples that could mimic I'm thinking of like 2D co-culture
Yeah,
experiments or 3D spheroid
I
cells.
think we're seeing it in like organ models.
Exactly. Organ model. Just thinking that something
like that.
Just millimeter, thinking of spheroid. Yeah, that's something you're thinking about. Yeah, that's cool. Alrighty. Yeah. Oh, thank you so much. Have cough, but mostly loves. Just hearts and watching. Thank you, guys. This is Ginkgo. I'm going to put these two streams together and show people. Yeah, all we had was 10 across platform. All right. Thank you so much. Let's say thank you.
Bye. Great to meet everyone.
All right. Take care. Oh, the